Dr. Knowles is here and is going to provide a bit of a background that Dr. McDally would, and then share his own slides. Thank you.
Hi, good morning. Some of you may know me as a cell biologist, but I'm actually a psychiatrist. And so I'm going to talk about some of my research in schizophrenia and touch into OCD. Is it not working? Or I got to stand into it. Okay. So Tim was supposed to talk in front of me, and then he would tell you what schizophrenia is, and then a few things about the genetic epidemiology at all. And the presentation seems to be lost. So schizophrenia is a disorder of thought where essentially you have a trouble distinguishing reality from non-reality. There's a whole bunch of other symptoms about evolution and hedonia and a whole bunch of other things about it. And it's thought to be quite genetic, 60-70% genetic. And so the largest efforts really in psychiatric genetics are to kind of look for the genetics of schizophrenia and what underlies that at the genomic level, and then with the hope that that will eventually translate into kind of understanding how schizophrenia occurs in the brain, and then we can have rational treatments. Tim would have also talked about kind of GWAS studies. I'm going to show a slide from a GWAS study, and when I get there, I'll explain it a little bit. I was hoping Tim would already explain that. And then there's a large degree that we're doing these GWAS studies in European populations, typically around the world, most of the GWAS studies for every disorder. And there's a great recognition now that we need to do these in other populations. So Downstate's actually the leader in doing this in schizophrenia GWAS is in African Americans and Hispanics, and then I also have a South Asian project I'll talk about in a second. So with that preamble, I'm going to go into my talk, which is, we actually know a lot about the genetics of schizophrenia now over the last 20 or 30 years. So we know variations, like a SNP, a one base pair change somewhere in a non-coding region of the genome increases risk. And that's great. That's foundational knowledge that won't change. It'll still be that way in 100 years. What we don't understand is how does that SNP increase risk? This is kind of the next major task of genomic biology and the genetics community, is to kind of figure out how do we go from these locations in the genome, which I call SNP space, to kind of understanding the disorder. And so this is kind of my roadmap, how I'm going about it. Everything that all the information that's coded for risk or protection in the genome is has to pass through an RNA molecule to make it out into the real world. And by the real world, I mean the protein world. That's where we operate. But it all has to go through RNA. So I think the first thing we want to know is what RNA molecule does this genomic alteration change? And does it change the level? Does it change the timing of that expression? Does it change the cell type it's in? Does it change finer features like the alternate splicing of that RNA molecule, those sorts of things. And I call that gene space. And then because I'm not a proteomicist and proteins are hard to study, I kind of make a jump right to kind of brain space, and that these changes in these RNA molecules are going to change structure and function in the brain. What pathways in the brain do they change? What firing rates of those pathways, how those pathways are connected, these sorts of things. And then once we actually even know some of those, like, we have a genomic location that changes X, how does that actually influence behavior? And how does that happen dimensionally in the whole population? Each particular pathway in the brain, what sets of behaviors does that influence, and in what direction as you change those different connections and pathways up and down? That's kind of the overall kind of thought space I kind of work at. See if this works. Good. So I kind of approach this at kind of different levels of analysis. And so projects in genomics, projects in transcriptomics, projects in epigenomics, and then in brain behavior and structure. I'm going to talk about two of these areas today, and I meant to have blue at the bottom of the slide too, where it says BTBD. So I'm going to talk about that if there's time. Anyway, I have multiple projects in the genomics region. Like this is in some degree starting to be a solved problem. There was a paper, a GWAS paper, that was out in Nature last week. 278 loci are now known for schizophrenia at a significance of 5 times 10 to the minus 8th. So we might say, you know, we have a lot of loci. We don't really need to explore that more. But those are actually, that's a very Caucasian-specific sample. So we just finished collecting a sample of 17,084 people last month in Pakistan for the study of schizophrenia, a GWAS. And I'm writing my progress report yesterday. We've done 2,500 people with bipolar disorder out of 10,000 we're going to collect from Pakistan. And then Carlson, Michelle, and I also have collected 4,000 people for OCD. So this is ways to look at how does the genetic structure and architecture of the disorder differ across populations of the world. And one population is going to inform all the populations, so it's going to be very beneficial to do this. Another advantage of doing things in Pakistan is 60% of the population are first cousins, so there's actually a lot of recessive forms of diseases. Childhood recessive diseases are threefold higher in Pakistan than they are in the European countries. So we think, and we already have some preliminary data for this that's up on the MedArchive, where we found 32 new genes for autism in a sample of 300 autistic probands and their parents. The genomics efforts going along has been incredibly successful. Over all the diseases and traits of humankind, these GWAS studies have now identified something like 387,000 loci when I looked it up in the GWAS catalog last night. So it's been a remarkably successful endeavor, just hasn't taught us that much about biology yet. So, like I said, changes in the genome, we're going to have to change a transcriptome, so I'm going to talk about a transcriptome project, one of 3 that we do. And I'm going to launch into that. This is a kind of a, you can think of it a little bit of an analogy of IPS cell lines, but these are genetically unmodified cells that we get out of patient and controls superior turbinates in their nasal passages. So essentially we put some viscous lidocaine in their nose and take a pair of forceps and rip out a piece of tissue, and then put that in a dish, rinse it up, and then put matrigel over it, and then neuroprogenitors grow out of that matrigel. Clonally passage them in a way that we get pure cultures, and we have these from 300 patients, and anybody who wants to volunteer to be a control after that description is really brave. So we have these 300 cell lines, and we did RNA-seq on them, and we asked what did the properties of this RNA look like? And so the first thing we kind of asked was the RNA. This is a representation of the 300 cell lines in black in the center of the slide there, and then it's in a field of colored dots, and those colored dots are each an individual brain sample from a human brain, where the fetal side is over there in kind of red and yellow and orange. Then there's a gap, and that's birth. And then there's kind of the green and blues on the right, and that's up to age 40. So the left side of the plot is the first trimester, and the right side of the plot is age 40. But we didn't order them by age. We just put the data into a PCA and then plotted the results, and the samples sorted themselves by age. Because there's a developmental program for how our brain develops, and that age is the major variant, the major covariate of what the RNA expression looks like from all these samples. Right there, it's PC1. That's about 20% of the variance. And then our CNON samples that we got from adults map into the fetal side of gene expression. So this is like a Wayback Machine. So we can take a nasal biopsy from a patient and know what gene expression in the fetal brain was like for them. And that's important because we actually think a major component of schizophrenia happens during fetal development. There's long been a fetal hypothesis of schizophrenia, and this is a way to actually see what fetal gene expression would look like in an adult. Because you'll never know, if you get fetal samples, you'll never know which of those was going to develop schizophrenia. You might have some idea of risk from their genome, but you don't actually know. So this is a way to actually know what the fetal gene expression is like of an adult with schizophrenia. And this would be true for any adult onset disorder that you want to study the fetal properties of. So I think it's a nice model. It's genetically unmodified. They're $200 to make one of these cell lines. So we have 300 cell lines. People making iPSC cell lines, they do this, and it's $5,000 to make a cell line. So they don't have collections of 300, and you'll see later having big numbers helps you use big data technologies and statistical methods to pull signal out of data.
2 minutes.
The other thing that's really good is these cell lines are 0.98 correlated with everybody else in the data set. All 300 are about 0.98, 0.99 to one another. In an iPSC cell line, even if we make an iPSC cell line from me and we get some subclones, 3 subclones, they could be as bad as 0.6 to one another from the same person.
1.5 minutes.
Pardon?
1.5 minutes.
Okay, great. I thought I got extra time because of Tim.
You did.
So we can also look at the data from these, from the gene expression, and on the left-hand side is a plot of these people and their genomes, and they separate by race in kind of PCA space. But when we look at the expression data by race, there's no difference. It's a cloud of mixed colors there on the right-hand side, indicating that there's actually not racial differences in gene expression. So the things that make your genome separate into the different racial groups are not affecting your gene expression, which is actually quite key concept, actually. So, how do these data correspond to schizophrenia? And this is the GWAS that I mentioned that Tim would have explained better. This was 50,000 people with schizophrenia compared to the genetics of 50,000 people without schizophrenia. You get these peaks, and anywhere above the line, there's significant peaks, and the article last week had 278 of those. How does our data line up with those? We actually had 3 of our genes be right under one of these peaks, and one of those peaks was FOXO3. I'm going to talk a little bit more about. And the reason why, one of the reasons it's interesting is FOXO3 has also come up on the top line here as a significant peak for brain surface area, and increased FOXO3 causes decreased surface area, and that's what's seen in schizophrenia also. And it's also a hit for IQ. So this is obviously a very important molecule for brain development, and it's a transcription factor. So we actually, in our RNA study of 255 people with and without schizophrenia, we looked at the gene expression between those 2 sets of data from the cases and the controls, and we found 80 genes that were significant at an FDR level of 0.1. And then we clustered those data, and this is kind of using these bigger data technologies and statistical things. And you see this kind of orange cloud in the middle, and that's a cluster of genes that are all being kind of co-expressed together, and and it's kind of denoted by the blue bar up at the top. And when we dig into that, we find out that these are all genes that are involved in a Wnt signaling pathway, Wnt5A. And on the left-hand side is how we turn on Wnt5A, because Wnt5A is down at the bottom. And interestingly, that's turned on by leptin, which has things to do with adiposity we referred to earlier. It also has IL-6 turning on Wnt5A. There was a study a few weeks ago that came out in JAMA Psychiatry where the IL-6 came out as a major molecule for major depression and schizophrenia and the inflammatory hypothesis of schizophrenia. So we're tying together inflammatory hypothesis. So that turns on Wnt5A in these cells, and it's up in the schizophrenics. And on the right-hand side, it's like, how does that signal back? Because we just have this pure culture of cells, so they're kind of auto-signaling. And lo and behold, if you go down the kind of right, I don't know if we get the cursor here. Well, you can see that actually increased Wnt5A turns on increased FOXO3. So we kind of now this connection between this pathway that is up in schizophrenia and it's turning on FOXO3, and now we have a connection between FOXO3 being up and decreased brain surface area. And so we're kind of trying to make these connections from transcriptomic changes into brain structure and function. And then we can actually take the data another way and ask the question, like, how many of the people in our study actually had this Wnt5a subtype? And it's best seen in panel C to the right, and you can look at the kind of bar at the top of the hierarchical cluster plot, and there's a purple segment of that, and those people have the Wnt5a subtype, and that's about 20% of schizophrenics. You can see on the controls in panel B, a few of those have the Wnt5a subtype. They didn't get schizophrenia, but they're probably at risk for that. And then on panel A is when we cluster the data with everybody together, and now we get something like 40% of people are in this this risk Wnt5a subtype. And so we estimate somewhere between 20 and 40% of people with schizophrenia have a molecular subtype defined by increased Wnt5a signaling and increased FOXO3.
Jim, can you wrap it up?
I'll end there.
Thank you.
And I won't talk about the second half of the talk. That's what happens when you have 50 slides. Okay.